so salty that no fish or plants live in its mineral-rich waters. Some 1,300 feet below sea level, it is the lowest surface on Earth.
Basically, you see the layers.
You see literally the layers.
Yeah.
Thousands of years.
Yeah.
For over 70 years, the Dead Sea Scrolls were considered the untouchable treasure of biblical history. Scholars believed they were the sacred writings of an isolated desert cult, perfectly preserved to validate modern religion. But recently, elite molecular biologists extracted and sequenced ancient DNA directly from the holy parchment. The internet immediately exploded with sensational claims that the biological results were literally not from this Earth. While those extraterrestrial rumors were heavily distorted, the actual scientific reality is far more controversial. What the genetic code truly revealed did not just expose massive international museum frauds, it completely shattered biblical history. The not from this Earth misconception. In the sweltering summer of 2020, the highly prestigious scientific journal Cell formally published a landmark genetic study regarding the famous Dead Sea Scrolls. Almost overnight, a massive internet frenzy erupted. Sensationalist digital clickbait flooded social media feeds, intentionally distorting complex paleogenomic findings into wild, speculative theories about extraterrestrial intervention. Headlines screamed that the ancient manuscripts possessed biological origins that were not from this planet, feeding fringe narratives about ancient aliens and cosmic watchers descending upon ancient Judea. You would think the world's top molecular biologists had just announced the discovery of Martian artifacts hidden inside the arid desert caves. But the scientific reality was deeply grounded in terrestrial biology, and it was arguably much more fascinating than any science fiction story. The viral internet phrasing was a deliberate misinterpretation of a highly specific geographic and ecological finding. When the dedicated research team analyzed the genetic signatures on several critical scroll fragments, they concluded that the materials were not from this land. They were not talking about another planet entirely. They were referring exclusively to the hyper-arid, harsh desert environment of Qumran, the exact location where the scrolls had been hidden away for 2 millennia. To understand the sheer magnitude of this revelation, we must look at the unprecedented paleogenomic data itself. For decades, historians assumed these historic texts were written on the skins of local desert animals like sheep and goats, species uniquely adapted to survive the brutal conditions of the Dead Sea Basin. Instead, the sequenced ancient DNA revealed a spectacular bombshell. Key ancient manuscripts, including significant fragments of sacred prophetic texts, were actually produced entirely from the skins of domestic cattle. Think about the biological implications of a cow living near the Dead Sea for a second. Cattle are massive mammals with extreme metabolic and nutritional requirements. They demand thousands of gallons of fresh water and endless acres of lush, green grazing pastures just to survive and thrive. The Qumran Desert is a parched, unforgiving wasteland where the blistering sun scorches the earth and fresh drinking water is incredibly scarce. Attempting to naturally sustain a large herd of domestic cattle in such a punishing, hyper-arid environment is ecologically impossible. Because these vulnerable cows could never have survived in the parched Dead Sea Basin, the heavy leather parchments made from their hides had to come from somewhere else. The sacred texts were clearly manufactured in fertile, agrarian regions many miles away, perhaps near the bustling city of Jerusalem, and then physically transported across ancient Judea into the isolated desert caves for safekeeping. This single, profound DNA revelation completely shattered a rigid, 70-year-old academic consensus. Ever since Bedouin herders first stumbled upon the hidden desert caves in 1947, the overwhelming academic agreement assumed that the entire scroll library was manufactured locally. scholars painted a vivid picture of a strict, isolated, desert-dwelling sectarian community known as the Essenes, toiling away in a remote scriptorium to painstakingly produce every single text by hand. The new genomic data absolutely destroyed that old isolationist narrative. Instead of a closed-off cult producing texts in a vacuum, the undeniable physical evidence demonstrates a highly sophisticated system of regional trade, dynamic agricultural resource management, and the widespread physical migration of religious literature across the ancient world. These invaluable ancient documents were definitely not dropped down from the sky by advanced otherworldly beings, but their true biological journey across the vast terrestrial Judean landscape actively rewrites everything researchers thought they knew about early biblical history, ultimately proving that scientific reality is often much stranger than modern digital fiction. The unprecedented DNA extraction challenge. Retrieving viable genetic material from the incredibly fragile remains of the Dead Sea Scrolls was long considered an absolute biological impossibility. When molecular biologist Oded Rechavi, biblical scholar Noam Mizrahi, and paleogenomicist Matthias Jacobsen first teamed up, they faced a forensic nightmare. You might assume extracting ancient genomes is a standard laboratory procedure today, but doing so with these specific artifacts represented a monumental scientific hurdle. These processed animal skins had endured 2 millennia of brutal environmental punishment inside the Judean desert caves. Over 2,000 years, the parchment was subjected to extreme temperature fluctuations, shifting humidity, and relentless chemical breakdown. The ancient collagen had essentially gelatinized, severely compromising any intact endogenous biological material. But the severe ancient degradation was only half of the overwhelming problem. The real disaster happened in the modern era. Since their initial rediscovery in 1947, these priceless fragments were subjected to catastrophic mishandling. Imagine the chaotic scene for a moment. Nomadic Bedouins, unregulated antiquities dealers, and multiple generations of early archaeologists relentlessly sorted, assembled, and closely examined these delicate texts entirely with their bare hands. Nobody utilized protective gloves or basic contamination protocols back then, completely ignoring the severe long-term biological consequences of their actions upon the incredibly fragile historical texts. Decades of heavy human contact thoroughly saturated the porous ancient leather with fresh modern human sweat, skin cells, and saliva. The artifacts had essentially become a hopelessly tangled genetic soup. To bypass this massive contamination problem, the dedicated research team had to completely rethink their analytical approach. Traditional paleography relied heavily on studying the visible strokes of highly unstable carbon soot and delicate mineral inks, but those fading surface inks offer absolutely zero usable genetic data. Instead, the scientists targeted the fundamental biological fingerprint perfectly preserved within the animal skins themselves. And to successfully ensure they did not destroy these incredibly precious historical artifacts in the extraction process, they devised an ingenious, non-destructive sampling method. They carefully gathered the microscopic biological debris that naturally fell off the degrading parchment over time, a rare substance effectively known as scroll dust. Working directly with this incredibly minute sample material, the team deployed high-throughput next-generation sequencing flawlessly combined with highly advanced hybridization capture techniques. This cutting-edge molecular technology allowed them to accurately target 2 extremely distinct types of valuable genetic information. First, they meticulously sequenced the mitochondrial DNA, which perfectly tracks the deep maternal lineages of the original animals. And then they thoroughly mapped the complex nuclear DNA, which provides incredibly high-resolution signature data pointing straight to isolated individual animals, proving absolutely vital for solving the overall geographic mystery surrounding these enigmatic religious documents once and for all. But they still had to overcome that massive modern contamination before proceeding. To effectively separate the ancient animal genetics from the overwhelming modern human interference, the researchers specifically developed highly sophisticated bioinformatic and complex statistical tools. These powerful computational algorithms were intricately designed to mathematically map the newly sequenced genomes and instantly identify the glaring biological signatures of recent human contact. By carefully running these advanced mathematical algorithms, the dedicated team successfully identified and thoroughly filtered out decades of careless human handling, eventually isolating the highly degraded, short fragment endogenous animal genomes hiding underneath. This unparalleled molecular achievement ultimately allowed scientists to definitively match tiny, completely disparate manuscript fragments directly to the exact individual creatures they originally came from. The entire grueling process remains a breathtaking masterclass in forensic molecular biology, transforming discarded biological debris into an astoundingly reliable physical method for perfect manuscript reconstruction that will forever change how we study ancient history. The shocking biological discoveries. Prior to the integration of advanced paleogenomic testing, a massive blind spot dominated archaeological circles. Historians and codicologists universally operated under the comfortable assumption that the vast majority of the Dead Sea Scrolls were manufactured on the skins of local desert animals. They pictured massive herds of specific desert-adapted sheep and goats grazing near the Dead Sea Basin, perfectly suited to thrive in the punishing arid climate. The genetic reality completely decimated this long-standing academic theory. When the dedicated research team processed an initial batch of 26 carefully selected scroll fragments through next-generation sequencing, the raw data produced an absolute biological shockwave. The molecular laboratory results definitively proved that while 24 of these ancient fragments were indeed manufactured from sheepskin, exactly 2 fragments were composed entirely of bovine leather. 2 of these sacred, highly protected texts were definitively crafted from heavy cowhide, drastically altering the known historical record. Finding cowhide in a desert wasteland is stunning, but the 24 sheepskin samples delivered an equally fascinating biological twist. You might reasonably expect all these desert sheep to belong to one tightly knit local flock managed by the same isolated community. Instead, the complex genetic profiling of the parchment revealed massive, unprecedented genetic variation. These specific animals were absolutely not raised within the local desert neighborhood surrounding the Dead Sea. The molecular data proved that these specific sheep belonged to highly diverse, genetically distant lineages. The animal hides used to manufacture these sacred parchments were sourced from countless diverse flocks scattered across vastly different geographic regions of ancient Judea. Think about the incredibly massive logistical network of trade and transportation required to successfully pull that off in antiquity. This extensive biological variation indicated a highly sophisticated, bustling ancient supply chain of raw materials, converging on the desert caves from miles away. By meticulously mapping these unique genetic signatures, the scientists unlocked a revolutionary new tool for precise historical reconstruction. They were no longer relying on human eyesight to piece together a massive, torn, ancient puzzle. The precise biological sorting allowed researchers to group microscopic, degraded fragments originating from the exact same physical animal. Imagine trying to assemble a massive jigsaw puzzle where all the pieces are completely blank, burned, and aggressively mixed with 10 other puzzles. That chaotic scenario is exactly what early scholars faced when staring blankly at thousands of tiny, unrecognizable scraps of fragile leather. The genomic fingerprinting completely bypassed human error, allowing the laboratory team to digitally reassemble heavily damaged manuscripts with unparalleled accuracy. They could definitively prove which tiny scraps belonged to the same original parchment roll by actively tracking the specific nuclear DNA of individual ancient animals. This stunning forensic precision exposed severe historical blunders made by previous generations of scholars. For several decades, traditional textual critics and early archaeologists had actively reconstructed manuscripts based entirely on superficial visual similarities. If two torn edges looked vaguely similar and the ink matched, scholars confidently glued them together in prestigious museum displays. The cutting-edge DNA technology corrected these historic errors. The advanced genetic mapping proved beyond a shadow of a doubt that earlier experts had mistakenly joined fragments from completely different animal species. In one spectacular museum blunder, past researchers had literally glued a piece of a sheep directly to a piece of a cow. They falsely assumed the text was one continuous historical document, based purely on handwriting and color degradation. This raw biological data forced embarrassed institutions to physically rip apart these artificial composite manuscripts, fundamentally changing the exact physical makeup of the ancient texts forever, challenging the Qumran origin story. For over 7 decades, a single untouchable narrative completely dominated biblical archaeology. The famous Qumran Essene theory confidently posited that a strict, severely isolated Jewish sect living in the immediate vicinity of the Dead Sea was solely responsible for writing and painstakingly maintaining the entire scroll collection. Scholars painted a highly romanticized picture of ascetic monks completely cut off from the outside world, endlessly copying ancient texts in a remote, dusty scriptorium. The newly discovered biological evidence absolutely annihilates this persistent historical myth. You simply cannot argue with the uncompromising laws of terrestrial ecology. When scientists definitively identified heavy cowhide fragments buried among the sectarian texts, the old isolationist model instantly collapsed under the crushing weight of basic agricultural science. Think about the strict ecological requirements of domestic cattle. These massive animals absolutely require abundant fresh water and endless lush grazing pastures to survive. Trying to raise a thirsty bovine herd in the hyper-arid, parched wasteland of the Dead Sea Basin is biologically impossible. You cannot sustain such large livestock in a barren desert. Consequently, these heavy cowhide scrolls had to be manufactured entirely elsewhere. They were undoubtedly processed in highly fertile agrarian regions, heavily favoring the lush Mediterranean climate zones surrounding the ancient city of Jerusalem or the sprawling Judean hills. After the scribes carefully inscribed these heavy imported parchments with prophetic biblical texts, someone physically transported them deep into the punishing desert. This stunning logistical reality forces us to embrace a drastically alternative historical model. The legendary Qumran caves were definitely not the exclusive, private library of an isolated ascetic cult. Instead, these remote, highly inaccessible caverns likely functioned as a massive, decentralized regional emergency archive. Picture the intense geopolitical terror sweeping across ancient Judea during the relentless Roman military advance. As heavily armored legions marched relentlessly across the landscape, eventually culminating in the brutal siege of Jerusalem, widespread panic gripped the population. Desperate Jewish communities from across the entire region frantically grabbed their most sacred, legally binding, and culturally significant literary libraries. They fled into the desolate wilderness, frantically hiding their priceless intellectual heritage inside these remote desert caves to protect them from total Roman annihilation. The terrifying approaching army systematically destroyed massive cultural centers without any mercy, forcing thousands of frightened refugees to permanently abandon their homes in a desperate bid for survival, while actively securing their sacred historical records from the impending absolute destruction ahead. This dramatic, decentralized archive model is strongly validated by an incredibly fascinating correlation between the genomic data and the ancient ink itself. Researchers did not just look at the animal DNA in a vacuum. They meticulously compared the biological signatures of the parchments against the specific scribal handwritings on the surface. The study revealed that scrolls exhibiting completely identical genetic signatures flawlessly matched highly specific writing traditions. When the parchment came from the exact same animal or a closely related regional flock, the text on that skin consistently featured the distinctive Qumran scribal practice. This is a massive revelation. This direct, undeniable link between genetic herds and specific scribal styles proves that highly localized scribal schools operated with dedicated batches of regional parchment. Scribes were buying their blank canvases from very specific localized herds. By tracking the maternal and nuclear DNA of the ancient livestock, modern science is literally mapping the physical geography of ancient text production. We are no longer just reading the surviving words of a vanished civilization. We are directly tracking their frantic, desperate movements across a war-torn landscape, following the unmistakable biological breadcrumbs they unknowingly left behind in the dark desert caves. The mystery of the book of Jeremiah. Perhaps the most theologically explosive finding of the entire biological study centers directly on those two highly anomalous bovine fragments. Both of these heavy cowhide pieces contained ancient writing from the exact same prophetic text, the critically important book of Jeremiah. For several decades, traditional textual scholars simply looked at the severely degraded leather, noted the highly similar handwriting style, and confidently assumed these fragments were merely torn pieces of the exact same damaged scroll. That basic assumption went completely unquestioned until the modern molecular biologists finally intervened. The highly sensitive nuclear DNA extracted from the ancient parchment revealed an undeniable biological truth. These specific fragments belong to two entirely different domestic cows imported from completely different fertile lineages, definitively proving they represented two completely separate historical manuscripts. Having two different copies of the same prophetic book in a massive regional library might seem utterly mundane at first glance, but when leading philologists deeply analyzed the actual Hebrew words written on these two genetically distinct cowhides, they uncovered a massive textual twist. The two manuscripts did not say the exact same thing at all. One bovine fragment aligned flawlessly with the longer Proto-Masoretic Hebrew tradition, the specific textual line that eventually became the exclusive, rigidly standardized basis for all modern Hebrew and English Old Testaments read today. The second cowhide fragment perfectly matched a distinctly shorter Hebrew text, the exact ancient version that served as the foundational basis for the famous Greek Septuagint translation. Think about the profound historical magnitude of that undeniable physical reality for a second. You have two vastly divergent versions of a deeply sacred prophetic text, carefully inscribed on costly imported cowhides from different agrarian landscapes, resting peacefully side by side inside the exact same desert cave. You would assume finding completely different versions of the Holy Scriptures stored in the same archive would represent an absolute religious scandal. For strict theological traditionalists, the radical idea of parallel, competing versions of divine scripture completely challenges the presumed textual stability of the ancient world. But this vital genomic evidence permanently shatters long-held academic timelines regarding strict biblical canonization. It definitively proves that multiple textually divergent versions of sacred scriptures freely circulated in parallel and were actively accepted simultaneously within Second Temple Jewish society. Modern religious traditions often demand absolute, letter-for-letter uniformity in their holy texts, treating any minor variation as unacceptable heresy. The biological evidence from Qumran unequivocally proves the ancient world operated entirely differently. This remarkable discovery completely reshapes our fundamental understanding of how the Hebrew Bible eventually stabilized over countless generations. For many modern believers and strict theologians, learning that their sacred history was once this unbelievably fluid and beautifully diverse can be a genuinely difficult historical pill to swallow. But the cold, hard molecular science absolutely refuses to lie to us. Ancient Jewish society clearly prioritized the broader theological themes and the overarching divine meaning of the prophecies over a single, rigidly standardized wording. The undeniable fact that an ancient librarian intentionally saved both the long and short versions of the book of Jeremiah indicates they viewed both textual traditions as entirely valid and highly authoritative records. Rather than representing the narrow, highly idiosyncratic views of a strict local desert cult obsessed with absolute ideological purity, the massive presence of these incredibly diverse imported texts fundamentally redefines the Qumran caves forever. The hidden desert library actually reflected widespread, mainstream, and highly fluid Jewish thought across the entire era, effectively preserving the vibrant theological diversity of a doomed civilization before the invading Roman legions arrived. Scholarly controversies and debates. You might assume that applying cutting-edge paleogenomics to ancient history would universally thrill the entire academic community without any hesitation. The reality on the ground was far more hostile and divided. The formal publication of the groundbreaking molecular data instantly ignited fierce scholarly controversies and intense geopolitical blowback across multiple disciplines. Traditional philologists and veteran paleographers aggressively pushed back against the highly celebrated scientific breakthrough. For these old-guard academics, the highly publicized DNA testing was merely an incredibly expensive redundancy. They strongly argued that traditional light microscopy of animal hair follicles and the extensive study of distinct orthographic variations had already clearly indicated that several manuscripts possessed outside geographical origins. Why spend massive amounts of vital grant money sequencing ancient nuclear genomes just to confirm what complex linguistics and basic optical magnification had already strongly suggested several decades prior? But the bitter academic infighting over laboratory budgets was absolutely nothing compared to the massive friction generated among theological traditionalists. For countless religious scholars, the definitive physical proof of textual non-standardization felt like a direct threat to their core belief systems. For centuries, conservative religious institutions maintained a rigid, uncompromising narrative regarding the absolute textual stability of divine scripture, insisting that holy texts were passed down through antiquity without a single altered letter. The undeniable biological evidence of parallel, competing versions of biblical books actively circulating together forcefully disrupts those long-held, highly comforting timelines of biblical canonization. It forces a deeply uncomfortable reevaluation of exactly how and when the Hebrew Bible actually stabilized into the singular authoritative version printed today. This highly controversial genomic revelation fundamentally shatters the long-established orthodox illusion of early singular biblical uniformity. Confronting the chaotic, fluid reality of ancient religious history remains deeply unsettling for those who demand absolute canonical perfection. Beyond the fierce theological debates, the physical custody and academic interpretation of the Dead Sea Scrolls remain deeply politicized on a massive global scale. These fragile, degrading animal skins are not just neutral historical artifacts quietly resting in secure, climate-controlled museum vaults away from the public eye. They are constantly and actively weaponized in highly volatile modern geopolitical arguments over territorial sovereignty, national cultural heritage, and intensely disputed historic land claims spanning across the West Bank and the heavily contested city of Jerusalem. Every single scientific revelation regarding exactly where these ancient Judean communities lived, traded, farmed, and wrote their sacred texts carries massive, undeniable political weight in the modern Middle East, frequently transforming routine archaeological laboratory discoveries into highly sensitive, fiercely debated international diplomatic incidents overnight. Finally, rigorous codicologists raised one absolutely critical caveat that threatens to fundamentally undermine the geographic premise of the genetic study entirely. They presented a massive, undeniable logical loophole in the main scientific conclusions. Identifying the exact fertile pasture where a specific domestic cow was raised and slaughtered does not definitively prove where the actual religious text was physically written. High-quality, meticulously prepared blank parchment was an incredibly valuable trade commodity throughout the bustling, ancient Mediterranean world. Think about a modern artist ordering an expensive premium canvas from France, but painting the actual masterpiece inside a tiny, cramped New York studio. A dedicated sectarian scribe working inside a harsh local Qumran scriptorium could have easily acquired a pristine, blank piece of imported cowhide from a wealthy traveling merchant along the bustling ancient trade routes. While the robust DNA data flawlessly maps the deep biological origins of the ancient livestock and their regional herds, the exact physical location of the ancient scribes themselves remains permanently shrouded in absolute historical mystery. A new chapter for ancient Judean history. This integration of advanced paleogenomics signals a massive paradigm shift in biblical archaeology, effectively transitioning the field from traditional translation into cutting-edge biocodicology and elite forensic crime fighting. Who could have ever predicted that decoding ancient history would eventually require heavily armed biological detectives hunting down the absolute truth? Consider the incredibly fascinating genetic case of a specific mystical text known as the Songs of the Sabbath Sacrifice. Researchers tracked a separate copy of this exact same manuscript found 55 kilometers south at the heavily fortified mountain stronghold of Masada. When they ran the molecular sequencing, the results revealed the Masada text was written on an entirely genetically distinct sheep lineage compared to the Qumran copies. This proves that deeply mystical religious traditions were absolutely not restricted to a single isolated desert cult. Distinct communities scattered across different parts of the country actively produced their own independent copies of popular religious literature, establishing a vibrant, interconnected religious landscape. But this advanced genetic matching provides an even more powerful, immediate real-world application. It serves as the ultimate high-tech weapon against rampant black market fraud. The lucrative antiquities trade is utterly infested with a highly sophisticated fakes designed to scam wealthy collectors and prestigious institutions out of millions of dollars. Think about that for a second. The spectacular bust involved a massive scandal surrounding the Museum of the Bible. Following their highly publicized opening, rigorous scientific testing exposed all 16 of their prized manuscript fragments acquired after 2002 as complete and utter modern forgeries. The brilliant forensic science deployed here is absolutely ruthless. Authentic ancient fragments exhibit a very specific biological signature. Over 2,000 punishing years in the desert, the natural animal collagen slowly breaks down into a distinctively gummy, glue-soaked gelatin. The forgers tried beating the system by painting artificial text directly onto genuinely old, completely blank Roman-era leather. They made a massive, highly detectable chemical mistake. To prepare the old leather for new ink, the criminals aggressively treated the rough animal skin with modern industrial lime to quickly remove the tough hair. This hasty chemical bath left behind massive, unnatural calcium spikes embedded deep inside the biological fibers. Under severe chemical and genetic analysis, these glaring mineral deposits glow like neon warning signs, instantly exposing the fraudulent historical artifacts for the incredibly expensive modern scams they truly are. Crucially, this relentless DNA analysis successfully resolved a highly disputed, decades-old classification error that had frustrated secular historians for generations. A deeply controversial legal document was allegedly recovered from the famous Cave 4 during the chaotic, completely unregulated Bedouin discoveries of the 1950s. For decades, highly respected experts heavily debated its true origins. The definitive genetic testing finally settled the bitter argument forever. The molecular profiling proved beyond any doubt that this specific legal fragment completely failed to cluster with any of the established Qumran manuscript genomes. It absolutely originated from an entirely different archaeological site in ancient Judea and was carelessly misattributed during the frantic, wildly lucrative antiquities market transactions of the mid-20th century. This final breakthrough powerfully demonstrates how modern interdisciplinary science continues to aggressively rewrite the social, economic, and religious history of ancient Judea. By seamlessly merging ancient history with cutting-edge genetics, we are no longer relying on educated guesses or superficial visual comparisons. We are systematically decoding the exact biological fabric of the ancient world itself, ensuring that the true historical origins of these priceless texts are permanently shielded from both innocent academic mistakes and deliberate modern deception.